Okay. Hey guys, it's an emergency. We have to convene a Farmer Bro Council immediately. Huge problems. New FDA Seaberhead. It's a big problem for Sarepta. Capricor is basically done. They're closing up shop. Anyone relying on anything but overall survival data is a wrap. Every single medicine has to prove an overall survival benefit from now on. 10-year carcinogenicity study. You don't have 10-year carc? Sorry, try again. NDA denied.
Order cancelled.
What's up, Luca?
Hey, Martin, how you doing?
Top.
Order cancelled.
Probably not what you were talking about. Sorry, I just hopped on here right now. But I was wondering if you knew anything about Lucid Diagnostics.
No,
That's not how
we're
you feel
talking.
about it?
No, we're talking about FDA.
Oh, sorry.
It's all right. It's all right.
All right,
It's just a bit of an emergency here.
yeah, no, I heard it earlier on the time. Well, sorry.
50,000 patient outcome studies for every single company. Retroactive. Yeah, that's what I'm hearing. That's what I'm hearing.
Order cancelled. Order cancelled.
You now have to do phase 4 clinical trials on your drugs that went generic 40 years ago.
Order cancelled.
What are we going to do, boys? I don't know what we're going to do. This is Armageddon. We wanted a rubber stamp. The hell is this? We wanted a rubber stamp. We needed a rubber stamp at FDA. Instead, they gave us this guy? Drake? What's going on? Quantavius.
Order filled. Order cancelled.
All pro drugs are now generic. No IP. 7-year orphan exclusivity is now 7-month orphan exclusivity. Sorry.
Order cancelled.
I don't make the rules. Vinay does. 2-year toxicology for antibodies now.
Order cancelled.
It's over, boys. It's over. Wrap it up.
Order cancelled.
Wrap it up. Just close all those biotech startups too. That's right. It was a good run. Biotech was a cool industry. It's over. Pull the plug.
Order filled.
Vinay, is Vinay FDA commissioner too? Is that just in? Vinay is the new commish. 10 minutes into the job, he's been promoted. Oh, fuck.
Order cancelled.
All p-values, am I reading this right? All p-values must be below 0.001? What?
Order cancelled. Order cancelled.
Two-sided? Okay, no more non-inferiority studies. Non-inferiority as a concept is gone. New p-value is 0.001. 10-year CARC study before you submit an NDA or BLA. No impurities. Less than 0.01% must be characterized with their own tox study. What else do we got on this list here? Just reading off the new things Vinay's going to do.
Filled.
Overall study for every single drug. Outcomes for every single drug. No hard outcomes, just survival. That's it. There's no blood pressure. There's no biomarkers. It's survival or else. I don't care what the disease is. Can't improve survival, it's over.
Order cancelled. Order cancelled.
The last thing on the list is to buy Pharma coin. Everybody buy Pharma coin. We're CTOing Big Pharma.
Yeah, that would be
Order
retarded.
cancelled.
That's retard stuff. We don't do retard stuff here.
Everybody.
No, you're a retard. If we could short that, I'd short that. That'd be cool.
Order cancelled.
Anyway, I don't know. Maybe it's
Order cancelled.
I don't know. I think it's probably much ado about nothing. You know, it's one thing is that I would say that Sarepta is probably more powerful than the CBER director. As crazy as that sounds.
Order partially filled. Order cancelled. Order filled. Order cancelled.
Crypto people, we're doing real business here, okay?
Order cancelled.
We get back to your games next week. This is real business, real industry. Factories and stuff. It's crazy. Actual stuff being made.
Order partially filled. Order filled.
Yeah, so biopharma undergoing some turmoil here. Share prices falling off a cliff as a new CBER director is
Order
announced.
cancelled. Order filled.
All approvals now personally will be approved by RFK. I don't know. Anybody want to say something? I did my best Kramer impression, but nobody's like Jim.
Order cancelled. Order filled. Order cancelled. Order cancelled.
Okay, again, maybe we caught the bottom of Sarepta. Maybe 47.
Order cancelled.
Maybe we caught the bottom. I was probably overreacting a little bit earlier.
Order filled. Order filled.
It's possible that the drug industry will continue. It's possible. Hearing reports that the death of the drug industry may have been greatly exaggerated. We may be back.
Order cancelled. Order cancelled. Order cancelled.
Oh, I'm also being told there is a new disease list where if you're not developing a drug for that disease, the FDA will not entertain any IND submissions, NDA submissions, any addendums to any open filing at FDA. Have to be on the disease list. The disease list has 12 diseases. They're all oncology. Sorry. I don't know. We'll see. I don't think it's all over. But certainly the drug industry has had its share of, I don't know, beatdowns. So it'd be nice to have a little relief. You know, you may argue that the FDA was a little too kind to Sarepta in the past.
Order cancelled.
You know, but it would be unprecedented to reverse an approval. I think that's out of the question. You know, you can't just do that as a one person opinion. You know, you need pretty broad scientific consensus
Order
to
cancelled.
do something like that.
Order filled.
So I doubt there'd be any drug approvals reversals. Some people have asked about the reversals of the past. One comes to mind called Zygris. I almost bought Zygris from Eli Lilly actually, and Zygris was this sepsis drug that had this like pretty specious phase 3 package, and another study was conducted such that
Order
it was, you know, sort of beyond a shadow of a doubt that the drug did not work.
Order
And the FDA removed Xigris from the market. There are other drugs like this. Trazolol was another one, if I recall correctly. And then one I was heavily involved in before approval, where I warned FDA not to approve it, was Belviq or lorcaserin. That story's been told somewhere else, but
Order cancelled.
lorcaserin got FDA approval and then was removed again by FDA. So,
Order cancelled.
you know, it's three drug removals in, what is that? How many years is that, guys? 25 years? Then you've got a couple of like liver damage issues that came out of the blue. So, for example, Baycol. Baycol was taken off the market for rhabdomyolysis. And then what else? There was another one. Ooh, this is my Encyclopedia of Pharma knowledge failing.
Order cancelled.
It was Resulin.
Order filled.
Resulin, an old diabetes drug that had a little bit of a
Cancelled.
safety problem. So it happens, but there has to be an exigent safety problem that's not one patient. That's, and usually it'll be something like, okay, we didn't know that the liver toxicity of this drug was cumulative. Well, the thing about Sarepta's drug is it's a one-time drug, so it's hard for me to imagine the one-time drug having cumulative tox
Order
since
cancelled.
you take it once. But you could miss it. Let's say you do a 12-week, diabetes study, and you have a small molecule sulfonylurea or something like that, and it's a chemical class known to cause liver enzyme elevation. So you give that drug, and over 12 weeks, you see a tiny bit of LFT elevation or something, but it's not significant. And then, at 2 years, you realize that it's actually a huge problem. That kind of thing happens, and that's when a drug could get removed from the market. But I don't think you could remove a drug that's been traditionally approved. Accelerated approval is a little different. Even those haven't been removed. So a good example is Provenge. Provenge is a drug that's sort of widely thought to not work. That got approval. So Provenge was never removed. And then, of course, interestingly, the other drugs from Sarepta, the PMOs, are generally thought to not work either. So you sort of have this issue
Order
of,
filled.
well,
Order cancelled.
you sort of have a company that's built on drugs that are generally not thought to work. And I think, the time to be worried about this would be if Sarepta was trying to get approval now. They already have approval for all these drugs. So, even though this guy seems to hate Sarepta, don't forget Sarepta has a lot of fans in the regulatory DC world. Way back when I was involved with AVI Biopharma in the early days of Sarepta, I was a little more involved. I tried to make some
Cancelled.
connections and inroads with a handful of very few people I knew that could be useful. And, I knew the company would need FDA on their side. And, they got approval of eteplirsen, their first ever exon skipper. Many people were against this. But, you were dealing with patients that were dying boys, Duchenne muscular dystrophy patients. So, if you're going to err on the side of somebody, you would err on the side of the kids. So they've gotten a lot of drugs approved this way. And,
Order filled.
I don't know. I think Capricor, for example, is trying to do a DMD drug with 8 patients. They did about a 22 patient study, but 8 patients got drug. 15 or so got placebo.
Filled.
It's pretty hard to approve any drug like that. And they're going for approval. They're getting an AdCom. And now, with this new guy in charge, I'd say that the odds of approval are basically zero. They weren't very good previously. Now they have to be zero. Sarepta is a different situation. All their drugs are already approved. So they have to go through quite a lot to actually kind of try to hurt Sarepta. It would be really
Cancelled.
long and belabored, kind of process, I
Order
think.
filled.
So I think that the Humacyte approval was also surprising to me. They might reverse that. that was a BLA, even though it's sort of a device more than a drug. And it certainly didn't meet a primary endpoint of any kind. So, the Humacyte reviewer was against approval. Um, so it was very, very kind of odd that they got approval somehow. So there are, I'd say 1 out of 100 or something like that. Maybe I'll leave it at that. 1 out of 100 FDA approvals are a little squirrely. There's some politics involved, but I'd say 99 out of the other 100 are legit, actual kind of rigorous science and legitimate and careful consideration of the facts. Pretty important stuff. uh, and look, every regulator is allowed to have an opinion, right? There's no problem with one person being more hawkish or dovish, to borrow a term from economics, or conservative or aggressive, maybe. And FDA has been on this aggressive side. They and a lot of investors I talked to thought they would become more aggressive. But what we're seeing is a huge, anti-pharma kind of conservative movement now, possibly, where FDA, could get very, very stringent, really digging into the 50, well, the title, I guess it's Title 18, 502, 503, all those rules where it's like, look, you need multiple well-controlled studies for FDA approval that all meet their primary endpoint, and if they want to stand on ceremony, they can do that. They have the full right to say no, that, new NDAs, we want to see 3 or 4 phase 3s work first. And, as I've said a couple of times, we're sort of at the end of medicine. You have people talking about longevity, which is not my favorite thing to talk about, because we still have medicine. When I say we're at the end of medicine, I'm being a little bit metaphorical. But I think we can see the light at the end of the tunnel of medicine, because all of our needs have been met. Many of our needs have been met on the primary care side. We have huge numbers of antibiotics. There's no point in developing antibiotics anymore. If you've tried, you've gone bankrupt. We have great medicines for cardiovascular disease, including the good old medicine of exercise. We have wonderful drugs for oncology that are, we're just starting on oncology. We're already cracked quite a lot of it, and that'll continue. We have 20 drugs for MS and other autoimmune diseases like Crohn's and rheumatoid arthritis. We're not quite all the way there yet, even on autoimmune, but we're getting there. So where do we go with medicine? I mean, there's pockets, right? There's Alzheimer's, there's Parkinson's, there's some genetic diseases, but even in the genetic disease toolbox, we have tons of options, right? We have tons of little tools that we can throw at these diseases. So what does pharma look like in 2040 and 2050 and 2060? We had this huge rush in the 90s. That was kind of the pharma small molecule glory days. And in the background, biotech was having its own little, coming out party. And then antibodies kind of took over for the 2000 to present day, basically, era. But now what? The multiple myeloma had no FDA-approved drugs. And the IMiDs came around. Proteasome inhibitors were the first approvals, and we don't even use them that much anymore. They're not as potent as a CD38, CAR T. You got 20, 30 choices for multiple myeloma. It's nuts. Oh, what's up, Parm? Good to see you. Haven't seen you in a while.
Yeah, good to see you as well. So increasingly, our company has gotten more in the side of doing small molecule discovery using our analytics as well. And what I find very interesting is that as longevity estimates increase, you have more complex diseases popping up, in the sense of, at least there's these high-hanging fruit, so to speak, that always seem to exist and roll over. And I think that that's something we should consider is, for instance, longer term off-target effects, like with the hydralazine. There was off-target effects that caused lupus for some women. And so what do you think about how that could kind of trail over, causing us to have more and more diseases to deal with by 2050.
Yeah, that's possible. I mean, a lot of people talk about post-finasteride syndrome, um, fluoroquinolones as well. A lot of people think if you take, um, what are they, the azoles, for fungal infection, that you have this like long-term side effect package too. So small molecules are just kind of, they're pretty random, right? They they're really not specific. They come from a different era. They of course can do great things. They get into tiny spaces that large molecules can't get to.
Being the big one,
What's that?
the blood-brain
Yeah.
barrier.
Usually the BBB is the main one of the main benefits of a small molecule, but you have others, and you have liabilities like you have low half life and low distribution usually because that's so small it gets filtered. So you got your pros and cons of small molecules, but by far the biggest problem and drawback of small molecules is lack of specificity. And I used to run all these like panels, just screening in random kinases just to see if you might get a hit. And I've increasingly thought, wait, why don't we do this more in silico? But regardless, it's 20,000 plus proteins. The chances that your small molecule might affect an important protein is non-zero, to say the least. So there's a lot of chemical matter out there. Thankfully, there are like certain hints and certain like things we've learned over the years of what to avoid metabolically, or maybe like from a certain scaffold or chemotype perspective. Some certain problem children in the chemical world seem to come over and over again. But yeah, if you could do a large molecule, I don't see why you wouldn't. Um,
Our bet is more so on like our ability to sense. So we're using like hyperspectral techniques to image cells having interactions with these molecules so that we can kind of understand what's happening in a more sort of live cell way,
yeah,
and like hopefully extend to organoids and so on and so forth. So
I like it.
we're hoping that works. Yeah, specifically we're focused on GBM, where it's like, there's that's our personal focus, but yeah, like that's the service as well. We want to maybe try to work with other companies
GBM, like
on, because we have our own, yeah, glioblast.
GBM. Okay, yeah. Interesting. Yeah, I mean, it's funny when you develop. Every time I think about developing drugs, I think about it in the abstract because like you don't have tools like you're describing. Like there's no visual tools. There's no like you don't get to look at anything. You just get readouts of assays. And
Yeah, we're getting terabytes of data per cell, but our throughput's low. But because we raised like $1 million, that's like not enough to like do much in biology. But our throughput's low, but our readouts terabytes of data that we're receiving.
yeah, I know some people who'd probably be interested. There's a lot of people out there who've made so much money in whatever industry, and they just want to invest in something that they think could help people. And GBM's so terrible. I happen to know like three or four people whose family, usually parents, have died of GBM, and it's just such a. My father died, as you may know, like 2 years ago, or actually a year ago. Has it really been that long? Um, and I guess it's almost 2 years now. It's crazy. Anyway, he went suddenly, and we didn't get a chance to say goodbye. GBM, they're all everything is kind of grief is terrible in general, but GBM, the victims or the families of the GBM patients often say that they got a chance. They get 6 months, 12 months to sort of it's a fatal diagnosis in essence, but there is this sort of small opportunity to spend time with your loved one or not. And I don't know, that's either that's consoling or not, but it's such a brutal disease that has withstood, you know, quite a potent effort against it in pharma, and still has very low
Order cancelled.
overall survival as far as I know.
Order cancelled.
Yeah, and it's all related to methylation, which is what I was studying. And, it is really sad, but also it is very interesting because it's related to the brain. And it
Order
seems
cancelled.
that there's a mix of detection and treatment that go with it. Like two of my cousins had it, had glioma, sorry.
Order
And,
cancelled.
unique sequencing,
Order filled.
like something that could work at a microscopic level, make a chemical
Order
map for you.
cancelled.
That's hopefully going to do something. But
cancelled.
these bets are like 10 years they take, and it's like every day is just wondering whether you're doing the right thing. So it's, hopefully
Order
someone can
cancelled.
benefit from this.
Yeah, I've been following this company. Dawn is the stock ticker. The company's called Day One. They have a BRAF inhibitor for low grade glioma, pediatric low grade. So, but it's like you're talking about a couple hundred patients at that point, and we got everyone's favorite boogeyman, which is you got 200 patients to treat. How the hell are you gonna sustain a whole company on that? Well, there is a way.
Order cancelled.
It's called having an extremely high price, and they interestingly they priced their drug about half of what I would have expected, and it seems like a very poor decision because if this community of people really is relying on you to advance
Order
care,
cancelled.
you're going to need capital, and the United Healthcare's and Aetna's and CVS's don't give a shit, to pay 400K or 200K or even $1 million for a drug. They do it all the time anyway. So it's a space that we've seen this in MS and myeloma. The more money pharma makes, the better the drugs that come out. And glioma and GBM both have been like these spaces where nobody's ever made a dollar, and it's kind of like your Pfizer's and Merck's and Novartis's are like, why should we do it? There's no money here. Let's do something else. And brain cancer is a real thing, and it's something that deserves more investment.
I'm so sorry that there was a cut, so I thought you were done speaking. But I was just saying, isn't it the case that some of this is because your discovery companies are relying entirely on these royalties to make money? Because it's like, for me, the first thing I thought, and maybe it's just my naive eyes, it's just been a year since we started, but immediately thought, why aren't these companies, for instance, also working as a service or as a diagnostic? That would actually make a lot of sense, right? Because they would get money from both places and be able to price more fairly because it costs less in total.
Yeah, I mean the pharma industry is just way larger than anything in the med tech or life sciences space. But for the longest time, these companies did have multiple streams of revenue and businesses. Roche still has what is basically the world's largest diagnostics business inside of it, and then Johnson and Johnson for the longest time had all kinds of things. They divested their diagnostics business a long time ago, but they still have a big med tech business. And occasionally, like Genzyme used to do a lot of diagnoses and different efforts to get the diagnosis out. I had a drug that had 60 people who took it, and it was such a terrible illness that if you found the patient before onset of illness progressed, you could really save their life. But if you found them too late, it was a permanent sort of neurological irreversible damage. So in that case, we invested a lot to try to get the word out, try to find these people before it was too late. So I do think drug companies operate in that continuum of education and diagnostics occasionally, where there's obviously companion diagnostics for EGFR and other mutations. Now I think more labs just do them, but back in the day you used to have to do them yourself. There's Genentech worked on fish assays for HER2 with partners. So I think the problem with, as with brain cancer is you just have like I said, I think there's a limited investment because there's so few people that have it. So there's just not that much attraction. And this company, Day One, I don't think they did anybody any favors by kind of pricing their drug too low, because if they had $1 billion drug, every big pharma would wake up and say, oh, we got to do something in this disease too. And before you know it, the 6-month survival becomes 24-month survival. And that's certainly what happened in oncology. In fact, oncology used to be considered a non-starter in pharma. Now it's actually the largest, revenue sector in pharma. But in the dawn of Taxol, one of the first chemos, and some of the other first chemos like carboplatin and cisplatin, basically they were really poor sellers, and pharma didn't think about pricing back then the way it thinks about it now. And they basically said there's no business here. So few people get cancer compared to diabetes or something like that. So, I think, of course, the extreme, version of this is one patient with one disease gets a drug made just for them. And, that's kind of breaks the whole system we have. We don't have a system equipped for that. But, someday. Have you been keeping up with what Kathy and Dr. He are doing? Did you see that? They got married?
Oh my God. Yes. But I, by the way, have to drop in 3 minutes. So, no, but thank you so much. And I mean, I always learn so much from you. But I was going to say, I absolutely have, I don't know if you've seen this, but I kind of went viral because I was like beefing with them.
Oh, that's funny.
I took pictures in our lab.
Oh yes, I saw that. It was so funny.
Yeah. No, I was just like, I didn't know this at the time, but like, he's with a teal fellow, which is kind of crazy.
Kathy's really sweet. She's one of the nicest people out there, and I'm sort of shocked they're married, but.
Like our investors are the founders of that fellowship, the Teal Fellowship.
Yeah.
And I didn't know that I was beefing with them. And so, like, it was kind of sad, but I mean, I just thought, they found it really funny, but I personally have a problem with economic gene editing because I think that life is not static, and if I thought that, then I would not be making non-destructive imaging methods. Because I don't know, I think it's like a very big bet to take, and I guess the one place where it's actually important to consider it is something like Alzheimer's, but didn't that guy just use it to protect emails against HIV?
Yeah, CCR5. You could do it with ApoE with Alzheimer's. It would reduce Alzheimer's by like 90%. It's just, Rubicon we don't want to cross, right? It's too scary. It's too dangerous. We were playing God, and that's frightening. And, they put the guy in jail for, just practicing medicine, in essence, with informed consent. I don't like that they put him in jail. The ethics of embryonic gene editing are probably past my pay grade and everybody else's. It's a really something that as a society we have to just get comfortable with and understand, and not go down the eugenics path, ideally.
Or just do epigenetic editing, like what's needed for most complex diseases.
Yeah, that might be a better way to do things, but I don't. I think that for therapeutic indication where the patient consents, the doctor consents, the state saying, no, we know what's better for your health than you and the doctor. Yeah, I don't know that that's really the way we should be going. This wasn't sort of like a situation where, they could just, it wasn't a cosmetic defect or something. It wasn't elective. It was pretty exigent situation. So, I don't know. I support Dr. He, and again, still knowing Kathy for 10 years, shocked that they're married, but happy for them. See you later. Okay, I think that's it. Yeah, so new FDA commissioner or CBER director, changing p-value. New p-value will be 0.01 going forward. 0.001, actually. Maybe even more zeros will be added by the year. 10-year CARC study required for FDA approval. 2 species. First time ever, 2 species. I'm just joking, of course, but the market's reacting at least as if medicine's over. And I don't know. Maybe it is.
Hey, Martin.
Hey.
I was just wondering, in terms of developing a new drug, what would you say the biggest price bottleneck or CapEx bottleneck would be? Like, what's the part that requires the most amount of capital? It's not actually developing the drug itself, is it?
Yeah, discovery is pretty simple. It's the clinical trials that cost a lot. So I'm bearish on all the AI drug companies because it doesn't really add up. If you've ever developed a drug, it's the 5 years of clinical trials you do, and the waiting and the enrolling, and doctors and hospitals and medical centers are very financially driven. If you're going to do a clinical trial, you better pay up, and then we'll do your trial. If you're not going to pay up, well, we got other things to do, and that makes clinical trials really expensive and time consuming.
Okay, so discovery is relatively cheap. So I'm assuming that also includes testing on animal models. It's cheap as well. So the issue is translating it over from animals to humans. What would you say the conversion rate would be between the two in terms of success?
I think that you sort of take the sort of a staged approach. Like, lots of phase 3s succeed because the company has de-risked it in sort of phase 2. So if you know that, for example, your blood pressure drug lowers blood pressure in phase 2, you can be pretty confident that it'll do so as well in phase 3. But sometimes you're really rolling the dice. So I think the translation is like, stronger every step you get closer. There are some animal models that translate with high fidelity to humans. There's some that don't. Generally anything involving the brain has a pretty poor translation to humans. But a lot of stuff that's like very genetic and metabolic, genetics sort of oriented is fairly good translation. Oncology is sort of notoriously poor translation. So it's a crapshoot. But I think the human clinical trials sometimes cost and take a lot of time because they are very long term. So that long term nature results in, like, let's say you're taking a disease that takes 5 or 10 years to sort of advance. You're going to have to do a 5-year clinical trial, and it's really expensive and it takes a lot of time. Your patent life is ticking away, by the way, as you do this clinical trial. Your patent life's starting to disappear. So if you're looking at Alzheimer's, which is possibly a 10, 20, 30-year disease process, right? How do you do a trial that's longer than your patent life? So I think there's a lot of issues when it comes to medicine that sort of have to get resolved. But the old sort of way of doing things is a little bit, it's not a fantastic system that we have necessarily. So I think the clinical trials are, far and away the most, kind of the most expensive part of the process.
Right, that makes sense. I agree with you. I would say if during the animal phase, if you can get an effect size that's very, very large, then in that case, almost always translates well into clinical. That could maybe remove the need for clinical trials. One more question. I know you're busy. But in terms of drug delivery, in terms of following the science, would you say there are any organs that we currently don't have good delivery mechanisms for, particularly in gene editing?
Yeah, just to take a step back, I don't think there will be a day where we don't do clinical trials. That seems like a really far away.
More so like much smaller scale. Like, say it works so well in animals and then you test it on a few humans.
Well, we have that already. I mean, we do have that already. There are many drugs that have gotten approved on a handful of patients, 10 patients, 20 patients. The results are so spectacular that they just get greenlit that fast. But yeah, in terms of delivery, I mean, we have nothing for the lung, the let's see, the bone is notoriously difficult. The muscle, any kind of skeletal muscle, cardiac muscle, are very difficult to deliver. Kidney, um, you know, bone marrow. These are all very neurological. Anything in the brain as well. Gene editing only seems to work, in fact, for the liver or maybe a local administration of, dermatological or, like a literal direct injection can sometimes work. But in general, systemic gene therapy has failed us, CRISPR and gene therapy, because of targeting. So gene therapy is a little different because you can get those caspids to sort of go to the right place if you try. But certainly on CRISPR, gene editing, we only have liver, and that's much ado about a really big invention in CRISPR, but no actual applications other than liver diseases, which already all these genetic liver diseases. It's a good time to have a genetic liver disease because you got 85 companies all going for the same three diseases: Wilson's, ATT or translate retin, and then what's the other one? The alpha one, antitrypsin. There's like 3 or 4 rare genetic liver diseases, and they're all being cured by 18 different companies. It's almost gotten hard to develop in that space because the drugs are too good. Like HAE is the other one, and all these companies have like the perfect drug. They're like, no, my drug's more perfect, and they're all curative basically. So we just love to get something in some other organ, and they do it in hematological, where they just blast the bone marrow and replace the stem cells, which isn't obviously really conducive to great medicine because you are, basically giving a stem cell transplant. So, that's sort of the state of the art. It hasn't changed much in 10 or 15 years, which is sad. And I think there was this hope that AAVs, adeno-associated viruses, would somehow have this tropism where you could engineer, the caspids to actually go to the place that you want it to go. And, that doesn't seem to have panned out. There was this huge investment from biopharma VCs and hedge funds to try to get these capsids and different AAV serotypes to go exactly to the right places. And really, we haven't seen anything. The only thing we've seen that's really big is the Avexis drug, which is now 10 years old or so. So, it's been a very, very slow, painful ride. And that's one of the reasons the biotech index, biotech stocks in general, a lot of people are sort of exhausted with a decade of, lost productivity, overcapitalization, all this stuff. And one of the biggest disappointments, I think, is gene therapy didn't carry some of its load that, we lost from other things. And it could have. Same thing with whether it's gene therapy or gene editing. But neither sort of became as promising as they could have been. And then we have all this other bad stuff in the backdrop that caused problems, like me, for starters. No, I'm kidding. But overcapitalization and chasing the same targets was a big one. But I think other parties, whether it's the opioid guys or whoever, didn't do any favors to the industry. But I think, hearing now that the FDA is going to get more conservative is certainly not something the pharma industry wants to hear.
Wait, I'm sorry. So when you say gene therapy, you're referring to like mRNA transient expression, and then gene editing, you're referring to more permanent, like actual DNA editing. And what would you
Not
say
exactly.
about nanoparticles in delivering mRNA? Do you see any hope there?
Yeah. So for gene therapy, I think when we say it in biopharma, what generally we mean is viral concatemeric kind of payload. It's semi-permanent. You get some transfection. It's not uniform. The concatemers, don't, the episomal, they're not.
Where would that transfection come from if you're delivering just RNA though?
Well, these are viruses, right? So they're AVs, and they're delivering DNA payloads. So they're basically forming these concatemers that stay in the cell and get translated, which is surprising. So they don't, for some period of time, there was a thought that they would actually be, be integrated, and there'd be a lot of concern there with, obviously, various issues. The payload for these things is pretty limited in size, but they basically stay as these little episomal, constructs in cells, and they read out decent amounts of viral copies. And if you have a disease that's like really, you just need a couple of enzymes to process some big fat, kind of clump. These are perfect. But if it's something where you need tons and tons of protein, you're probably not going to do great with AAV. Now, the mRNA is new, right? We have one sort of mRNA modality, which is the vaccine. And certainly the lipo nanoparticles and so forth seem to get you liver expression. But, when I was sitting around thinking about whether the Moderna vaccine would work, I was fairly convinced it would not work because big charged molecules don't just slide into cells, right? They tend to not do that. And, you need to be under 500 Daltons, you need to be nonpolar, you need to, it's all basic kind of chemistry. And, they got it through the LNP where they encapsulate, all this charged stuff inside the LNP. The LNP sneaks in to the liver cell, but it's not sneaking anywhere else in. So I think there's lots of ways to solve this. And I've tried so many myself, and it seems like damaging cell integrity isn't one of them. That's one I tried a couple of times. Like things like anthrax and HIV find ways to deliver payload into cells and keep the cells intact, but they tend to also actually be fairly toxic. So some people have tried to use those. I did a deal with sick kids, University of Toronto, to try to use the anthrax machinery to deliver a valuable payload. AAV is gentler. We get adenovirus infections all the time. We don't even notice. And if we do get them, sometimes if we notice them, we see them as a minor cold. So they're like the perfect way to deliver the payload, but it's just been hard, man. I mean, there's no easy way out of this. And unfortunately, again, this was supposed to be the next chapter of the biotech industry. Perfect gene editing or perfect gene delivery, whether it's, like you said, a really super transient RNA or this like semi-permanent AAV that everybody's using, including Sarepta's AAV levitus with microdystrophin for DMD patients. Or more permanent solution like CRISPR. There's those liver diseases that are being treated, but there's no obvious direction to go next for liver, other than liver. Well, I'm sure some enterprising companies will figure it out someday, and we'll have a biotech boom again, but probably not this decade, unfortunately.
Just to clarify, the AAV, it's only semi-permanent if it's carrying a DNA payload. If it carries RNA, it would be transient. Is that correct?
Well, my understanding is they are DNA viruses. So they're not, I don't...
But if they're not carrying DNA, only RNA, and we're just using the capsid as a way to get
right.
the actual RNA to the organ, and then it injects it, but injects just purely RNA, then there wouldn't be any concern over permanence.
That is true.
Martin, can I answer real quick for it?
Yeah, go ahead.
Real quick. So the only situation you're describing that leads to permanence is when there's a complementary version of that RNA that binds to it and creates a cDNA, a complementary DNA, which is two single RNA strands together. That would still allow it the molecular machinery potentially to hitch a ride on the types of proteins that transport DNA into mitochondria or to the nucleus. So there is a chance of permanence even with an mRNA administration.
Okay, and is that the concern that people are having about the mRNA vaccines potentially increasing risk for cancer?
Yeah, it's totally possible, but these are super low rates. I mean, it's already a small payload that depends on continued replication of it. And on top of that, to have a statistical chance of DNA formation and transport is another smaller chance, but it's technically possible, and there's a potential for it. And the only other point was about Gen Kui He. Yeah, you can't genetically engineer embryos because embryos eventually produce germ cells, which means like they produce a sperm or an egg eventually. And if they do that, that means that there's potential of carrying on that gene editing into generations in the future with unintended side effects. So that's why we have what we call the Weissman barrier, or that moratorium in the late 1970s. That isn't like state directed, but like a confederation of states directed, like World Health Organization level. I don't know exactly which organization, but it's an NGO essentially that made scientists agree not to do that. So that's just the other caveat there. All right, thanks. Bye.
Yeah, and when you talk about the barrier, or like I think the point is that the germ cells get the mutation, right? Are you talking about the machinery,
Yes, exactly.
the machinery coming with, or are you more talking about just the propagation of that?
The propagation of that. The machinery is not not going to come with, right?
Right. Okay, just making sure.
Right? But the point is, the propagation of it into haploid cells is definitely possible.
Oh, that's the point. Yeah.
Yeah, that's the key. That's the reason why we have a moratorium on it. That's why he went to jail for it.
But
it's not so much like, hey, these are consenting adults. It's like, bro, you're a scientist. You're supposed to know that you can't do that.
Well.
You're bad at your job. You know what I'm trying to say?
Well, just to put.
Surgeon operating on your brain and then being like, I'm practicing medicine. It's like, no, bro, you didn't know about that. Why are you doing that?
Well, just to push back a little bit, and I'm not saying that what he did was right or wrong, but just to push back a little bit, there's books about CRISPR, Nobel Prizes about CRISPR. Everyone's excited about CRISPR, and then a guy actually does it, and people get mad. it's kind of silly.
For sure, but I'm just saying, that, like, according to the grand scientific community, it's wrong because we all agree not to do that because of exactly that description, which is there's the Weissman barrier, permanent transfer to haploid cells without intended effects. So that means that you have such a vast lack of knowledge on the actual effects of what you're performing. That's like, I guess you have somebody that consented to that, but there are continued effects that ripple through humanity that are beyond that individual's consent.
Yeah. And again, we could also just call that natural genetic drift, right? Like every mutations happen.
I guess you could call it natural from the minute you push the first domino.
You might. Just at this very second, the right UV ray could be striking you, and changing a gamete,
for sure.
And you could have a child tomorrow with the exact, and it might, be more fit or less fit or.
Just a straight radiation. Yeah, I happen to walk past uranium ore. Exactly, same thing. We're constantly bombarded with genetic changes. There's no question. It's just we probably shouldn't do them to each other like that. Maybe other ways would be dope.
Yeah. No, I'm just playing a little devil's advocate. I don't know what's right or.
I appreciate that.
Yeah.
When you brought up Wilson's disease and all these liver diseases, the only thing I would elucidate is hey, they're all trying to go for low. You have to do is affect one protein, like one transporter, one alpha trypsin that you can outproduce the mutant version. It's all just trying to minimize the amount of additional complexity to showing it working. And I honestly think that's a space where large language models, once they get comprehensive enough, are going to be very helpful for us understanding more of the theoretical pathophysiology. So we're not just attacking these diseases that are singular proteins and outproducing them just by having a regulatory pathway that upregulates the production of it. You know what I mean? Maybe we'll eventually have serious ability to build a megazord in there. You know what I mean?
Yeah, no, I'm a big fan of this. Very few people know I actually started a little natural language processing company in 2015 because my bottleneck was reading PubMed. And, I was in this arms race with my friend and competitor Vivek, who hired a big team of people to do the same exact thing. And we kept kind of paying people to sit there and troll through PubMed. And there's 35 million articles in PubMed. So
let me pitch you something.
Let me just finish. I definitely want to talk about it, but let me finish the story. So it's sort of an NP complete problem, at least with humans. At the time, we brought IBM in, and they showed us Watson, and I asked Watson what the difference between type 1 and type 2 diabetes was. It didn't do very well, and the rest of the future happened. But I quit NLP. And then, Transformers came out. The woman who I'd marry, I at the time was studying them and was very early at a company called OpenAI. And now she's still there many years from now. And she was sort of the one person who knew anything about biology and medicine at the time. They were a very, very small company. So she pitched Bill Gates on some biology work that they did. And lo and behold, Microsoft did a deal with the company. So I think there's a lot of hope that LLMs can teach us a little bit about biology and teach us what lies beyond. Probably more in the, take the massive human knowledge that we built, 37 million disjointed papers, where the only person who could synthesize it all is some genius that can read really fast, which has, no matter how smart this person is, there's limits. Or an organization that could read really fast. And if you've ever been a part of a big medical university or company, that doesn't work very well. or a machine, and the machine seems to be by far the best, choice. So the question is, using these machines, can we get some really great insights? I like to give this example of the BTK inhibitor. The BTK was well known to be a really important hematopoietic enzyme in the 80s, and it took 20 years for somebody to say, let me make a BTK inhibitor, and countless people died of lymphoma that could have been saved. But for the connection of these 37 million papers, and forming these inferences, we could get there. And I think, whenever the people at OpenAI or any other company ask me how is AI going to help medicine, I basically say it's not. And they say, well, what about these drug discovery programs that are AI powered? And I'm like, it's all hocus pocus. What we really need is better targets and better strategies. The medicine's there. We have all the chemistry, we have all the biology. We just need ideas, and those ideas are the kind of the important part that moves medicine. Is somebody stands up and says, I think IL-4 blockade will be great in asthma, and you get a Dupixent. And other people say, I think this thing will be great in that, and they're wrong, and they spend a lot of money, and that sucks. But it's the idea generation that's the most important part of our industry. The chemists are important. I'm a chemist. The biologists are important. The assay people are important. The manufacturing people are important. But at the end of the day, unless somebody stands up and says, I have a therapeutic hypothesis, here it is. And up until now, the only therapeutic hypotheses that have ever been advanced have been by humans. And I did a fun thing with my girlfriend. We're technically not married yet, but regardless, baby boy coming in August. My girlfriend and I wrote a for loop where we fed GPT basically every single protein. We said, okay, 4i equals 1 to 20,000. She's a better programmer than me. What protein, when inhibited, what disease could this protein potentially, cure or alleviate or whatever. And you could run that for loop and just go through every single, theoretical, protein in the body and knock it down with an RNA or an antibody or small molecule or whatever. And, conceptually, that's kind of, all we've got to go on, right? Maybe there's medicine beyond proteins, for example, epigenetics, custom proteins, tons of different little modalities here and there. Certainly, epigenetics comes to mind, post-translational modifications, but, cell therapy, I don't know. But certainly starting with that and asking a super intelligence, I think Sam Altman talks a lot about, what would you do if you had the God machine? And, a lot of people say, gee, I don't know, I would solve the Riemann hypothesis. Well, as a math fan, I think that's a great idea. But as a humanist, which is about 1 minute of my day, every day around about 30 seconds, I have feelings. And I would try to think about maybe, would there be a good disease we could treat if you have the super brain, GPT-5 or GPT-6 or GPT-7 or whatever. Why not point the super brain at one of these diseases and say, super brain, what's the best way to treat Alzheimer's? And the super brain may say, I think the best way to treat Alzheimer's is it's really an autoimmune disease, and we should come up with an autoimmune modulator for the brain. And here's a couple of ideas. Here's a CXCR3 inhibitor. Here's a CD20, a small molecule. Here's a PD-1, small molecule protein-protein inhibitor, interaction inhibitor. And here you go, try these out. And 5 years later, you have the best Alzheimer's drug. So I think that's kind of the direction we're going. Anyway, I said a mouthful because I just like you, I have a lot to say and think about on this topic. I do think it's really exciting. But what do you think?
Wait, yeah, so you're saying idea generation and discovery is the most important part. I agree with you there. Would you say it's entirely possible, let's say for a single person, individual, they come up with the idea, would it be possible for them to execute it and actually create the drug and maybe test it on a mouse entirely by themselves?
Very rare.
With things like cars and rockets, you can't do that because you need, team of engineers.
I'd say it's easier. I'd say it's easier with cars and rockets. I think, yeah, I mean.
Single person, finding the target.
Okay, so let's.
And then getting the plasmid, transfect the cell, design the actual gene construct. I mean, there are companies that manufacture synthetic genes now.
The future is on chips. So what you're describing, if you were going to have a single person do something like that, it's eventually organ on chip or organism on chip approaches, not necessarily the live animal entirely. That's probably the most
Well,
I'm talking more so you test it on cell culture, and
sure.
let's say the effect size is so strong on cell culture, then you move to a mouse.
Oh, for sure. So
Then mouse.
yeah,
And then if the effect size is
right.
very, very large, then maybe a few humans.
Well,
you could do that if you're a doctor.
So the problem becomes, so if you're an MD, PhD, or something like that at a teaching hospital, maybe you can get, the wet lab time, or it's your lab, you could do it and you publish it. And this is exactly what Vivek and I would troll sort of thousands of papers to find the one random hospital in the Czech Republic that did a great job with, in fact, the drug tenofovir was invented in the Czech Republic, and Gilead bought it for $1 million and turned it into a $100 billion company and changed the face of HIV at the same time. So, the problem is, you publish that paper and nobody cares. You present it at a conference, nobody cares. And you sit there and, say, I need $10 million to do a real animal study or to file an IND. And, the university is saying, we don't have $10 million for you, and nobody else does either. And, you might get lucky and get one or two pharma tech transfer to pay the visit, but you don't get the check. We were competing with Johnson & Johnson to do with Memorial Sloan Kettering, the ketamine for treatment-resistant depression. And, that was like the greatest psychiatric advance in 30 years. And there was still just two companies kind of like half-heartedly kind of, interested in it. So it's tough, yeah.
So that's what you were doing with Vivek then? Basically, you're hiring interns and then essentially having them peruse through PubMed.
These were...
papers and looking for that one from the academic center that was actually real.
Vivek.
Real quick, I want to pitch you something, Martin, real quick, because it goes back to the idea generation thing, real quick.
I just wanted to answer that last point because I wanted to clarify. Vivek was a competitor. He was an investor in my first company, which did this. My second company as well was a sort of competitor. We didn't hire interns. We hired people, MD, PhD type people, paid them close to $1 million TC. So it was like, we had 20, 30 people each. And Pfizer and Novartis had like 5 people each. So we thought this was the biggest misunderstood thing in the world. And we still did think that, I think. But anyway, please go ahead.
So you know what a PICO question is, obviously.
Well, I know what PICO anything is, but what's a PICO question?
Yeah, like PICO, population intervention comparison outcome. It's
okay.
acronym using of a clinical question, right? So if you're going to do an evidence-based medicine approach, you come up with something called a PICO question. PICO question is like in, patients that have lymphoma versus don't have lymphoma, that's your population. Your intervention is a BTK inhibitor versus standard therapy. Your outcome is, whatever cell count you want to look at, for example. And that's your PICO question. You have an intervention, a comparison, and an outcome, basically. Does that make sense what I'm saying?
Sure. Sounds like a scientific method to me.
Yeah, it's our acronym for clinical question formation. So, back in that same time period where you're doing NLP in 2015, I'm doing something similar. What I came up with was an automated PICO question generation, where you, let's say we have all 37 million PubMed papers. You define every variable across all 37 million as a population variable, an intervention variable, comparison variable, or an outcome variable, and then form every single question that's permutatively possible. And then assign the statistical methodology that's appropriate for it, depending on whether the independent or dependent variable is categorical or ordinal. And then you do a Bonferroni correction to look for family-wise error rate and just surface whatever's statistically significant, and then cross-reference it for clinical significance based on minimum clinically important difference. And literally just squeeze all the juice out of the data set, out of the entire world data set as fast as humanly possible with the technique.
It's like GWAS. GWAS for GWAS for published work. Yeah.
Yes, you got it. And for registries and for large healthcare data sets. I pitched this to the White House in 2020, and it's exactly what you described. Everybody's like, "Oh, that's amazing," but we don't really care. No one really cares. How is anyone going to benefit politically from this? Politically, on the grand stage, but just
I'm a market guy, so at the end of the day, the easiest solution here is just Eli Lilly or Merck says, "Oh, we need to use this," and they write a check to my girlfriend's wonderful company, and before you know it, they use this tool or the rival or Google's tool or whatever, and they create the next new great drug for Parkinson's or whatever. And I think that's probably the best way it goes. And what's funny about this is, I remember when GPT-2 came out, and you could talk to it, and it was not great, but it was interesting. And basically OpenAI, didn't want to make ChatGPT. They basically they said, we're making an API. You can tap into it. We're not a product company. We're not here to make chatbots. We're not here to make software. We're here to just research AI. And then they said, all right, let's, nobody's building the tool that we expected. We gave you the API. I gave you everything on a silver platter. All you had to do was just connect it to like a HTML box. You would have had it. And they said, no one's doing this, so we're going to do it. And then they one of the most popular pieces of software ever. So they wanted to do it. But I think that the moral of the story is that, it might have to be one of these AI places that says, okay, we're going to have to do drug discovery. Because if you know the Mercks and Pfizers and you've ever worked at some of these places, things don't change quickly. Pharma, one of the reasons I don't want to be in the pharma industry anymore is it's a very stultifying place. People like Vivek are extremely big change makers that they're doing things way different. The pharma world is like very entitled and monopolistic, like we have a way of doing things, we're not going to change it. Vivek sort of turned a lot of things upside down. A lot of people didn't like it. And, it's just a very sleepy old culture. There's a lot of young people in tech. They do things very differently, move fast and break things. It's a very different world. You can't do that necessarily in pharma. But, this idea of saying, okay, how about a company where the central hypothesis of what drugs we advance is decided by AI is a really good idea. And I think that, your point of like, okay, how do you orchestrate this in this like interesting matrix, multi-dimensional matrix, is a really good way to do it too. I think you could do it either way and see which way works best. But I do think the idea of like somebody sitting at Biogen and saying, I really like neurology. I know a lot about neurology. I'm going to tackle this disease because I'm Mr. Neurology. And the problem is, you don't know if that guy's an alcoholic. You don't know if that guy's, has hubris. You don't know if that guy has, he's gotten one lucky drug right, so now he's got a huge budget, and he's going to get them all wrong. You sort of have human fallibility to deal with. And, this is the reason Wall Street was taken over by the quantitative investors. 9 out of the top 10 hedge funds are quants. And, the machines do a better job. And it's just very hard to deny. And, at some point, there's 8,000 drug companies. One of them is going to be run by a machine, and slowly but surely, it's going to. Well, of course, the humans will do the experiments, but slowly but surely, I think that the machine run drug company beats the human run drug company, just as the machine run hedge fund seems to beat the human run hedge fund. And again, the machine's a tool. It's not an adversary. It's not like some anthropomorphic like evil. It's just another tool that we do our job with, and that's there's nothing to be scared of for that.
Would you say that you and Vivek, that the big thing, like 99% of the gains, it came from that idea of hiring 20 to 30 MD PhDs, whereas these Pfizer's, whatever, they only had 5, and then having them full-time sifting through papers on PubMed, looking for that one gem from an individual researcher?
Yeah, the bid-ask spread basically was the average university or failed drug company or whatever wants $1 million for a drug, and that's the bid, and the ask for a working drug is like billions of dollars. And in fact, Vivek did a tremendous transaction where he bought a bunch of drugs from Pfizer for 100 mil and sold them to Roche less than a year later for $6 billion. So one man's trash was certainly another man's treasure. And we just did this over and over and over again. It wasn't hard. Pfizer had all these failed drugs, and Merck and Bristol and all these guys, and we took a drug from Bristol, my company, that we paid $2 million for, and we got FDA approval. So with a brand new disease, first ever in its class, et cetera. So it's just the biggest mispriced asset market. You look at stocks all day, you're trying to figure out if Pfizer's worth 24 or 25, but then you look at a drug and you're like, this thing could be worth $10 billion. And,
just to make sure I have the flow right,
yeah.
So the academic individual researcher, they discover, create the drug, and then they sell it to you guys for $1 million, and then you develop it a little bit further up to the point where it's maybe worth $100 million. So maybe you run phase 1 trial, and then you pass it off to Pfizer, and then they make you sell it to them for billions, and then they take it throughout phase 3 and onwards. Is that correct?
No, there's lots of different configurations. I mean, we took our drug all the way to FDA approval, and the company still sells it today. Vivek did that many times as well. Sometimes the pharmas never bite. They just never bite. And the other thing I'd correct is that it's not always some like small university or something. It's oftentimes a drug company that, for example, yeah, they like Amgen might just say we're not doing psychiatry anymore. They shut down 20 good psychiatry programs. They say we just decided not to do psychiatry. Sorry, and it's like anybody wants a psychiatry drug. Here you go.
Well, how do you know those buried assets exist if you're not in the company themselves?
Patents, trials, papers, word of mouth. You build a network. People that used to work at Amgen. Oh, this drug's coming up for license.
Okay, that makes sense. Let's say you find the drug, then what's the next? Let's say you find the buried asset. What's the next step after that?
I mean, you start a business. It's a business now.
Like practically speaking, what would you
depends.
do? Would you literally take the drug and test it more extensively in animals, or would you test it in a human?
It depends. Depends on what the pharma had done. If the pharma last left off, in a certain place, then whatever you need to do to get to the next value inflection point, whether that's a phase 3 or you got to go rework some toxicology, or you got to manufacture more drug, which is often so. In any event,
so you're just picking up the academics work and you're just taking it forward a little bit farther. Because the academic, they just don't have the resources to do it, and they can't communicate it well enough to the point where anyone would give them a chance.
Right, exactly. You need buy-in, and even it's funny because one of the dynamics that's really fun is that let's say there's a drug, let's take Viagra at Pfizer, and you do the drug, you develop the drug for cardiovascular disease, it doesn't look promising, you pull the plug. Let's say you're the guy, the program lead, you go to your director, or maybe even the CEO of the company, and you say, I want to do it for erectile dysfunction. And the CEO says, so let me get this straight. You just lost us $50 million on this cardiovascular thing, and you want to take this dog of a drug, and you're asking me for another $50 million. And it takes a lot of guts to be that person that says yes. Most people say, crawl in a hole and say, oh, I hope I don't get noticed because I lost $50 million. So sometimes some of these drugs have been in 4 or 5 autoimmune diseases, and it turns out that and they don't work for any of those, and then, oh wow, actually this is the best drug ever for Sjogren's. I hate that. I hate saying that thing. Or sarcoidosis or something like that. And there's no drugs for those diseases. And one of the reasons is just somebody at Roche or Pfizer or Merck doesn't want to say, I need another $25 million to do the Sjogren's. It's like SJOGRENS. It's like the hardest word in medicine to pronounce.
Like 25, 50 million, these are huge numbers. What do those expenses go to? Obviously, the reagents. If you were to break
clinical trials, they're all clinical trials.
Okay, but in terms of developing with the discovery process, just creating.
These drugs.
How expensive would you say that would be?
These drugs almost always already exist, so it's all clinical trials. But it's.
They exist and they've been tested on animals and everything.
Yeah, they've usually been tested on humans already too.
Okay, let's say for the clinical trials, what's the most expensive part there? Is it hiring the doctors to conduct the trials?
Yeah, CRO. The CRO does it for you, but the CRO goes and does all the paperwork and opens up every study site and pays the doctors and the site for doing the trial, collecting all the data, writing the protocol.
Does it have to be an MD, a doctor, to do the clinical trial, or can it be a nurse or nurse practitioner?
It's a very, very rigorous, process. Everything has to be documented very carefully.
Okay, so it's not just the MD, because I'm thinking about approaching this from first principles. Would it be enough to just have an MD and let's say they're well versed in all parts of the all parts of the process? Can they just handle it all themselves?
Well, you got to file an IND too, and you have to file something with the FDA, and the FDA usually gives you something called the study may proceed letter.
Right,
so,
but you can have AI do all of that paperwork.
Yeah, FDA usually wants to meet with you, kick the tires a little. It's not.
So say FDA wanted to meet, and I'm just one person, one MD. Is that convincing enough, or do they require a bunch of NPCs to be there?
Yeah, they don't mind having some NPCs. Again, that's why this industry is what it is. I mean, you have a very old way of doing things. Somebody like Vivek or myself shows up and says we're going to change everything. It's not something people like. It's something that people, again, it's a lot of old line thinking in this space. New thinking isn't necessarily that welcome. It's a very clubby oligopoly that hasn't grown very much.
Well, maybe with this administration, they'll be open to change.
Yeah, I think so.
One more question, I have to go afterwards. But in terms of your relationship with Vivek, so you guys were competitors back in the day. Did you ever work together?
Yeah, so he was in the hedge fund business. So was I. I started a drug company that he invested in, and the company did really well. And he left the hedge fund industry to start a drug company that did largely the same kind of stuff. I think we both shared this idea, and we weren't the only ones, by the way. I mean, there are many, many people in pharma that realize that the trash of many drug companies and universities is actually treasure.
So Vivek was at a hedge fund. He started this like bio thing, and then you invested in it. And then when it started,
Jesus Christ, no, backwards.
No.
So yeah, I started. Okay. That's right. He was at a hedge fund called QVT, and he was very good at what he did. I had wrapped up my hedge fund efforts and started a drug company. He invested in the drug company. It did very well.
Was it the first time you ever got into bio?
I'd been trading, investing biopharma companies for the last 15 years before then. But yeah, it was my first ever drug company. I was 28.
So you're trading bio companies at the hedge fund that you're working at full time, and then you started your own drug company.
Bingo.
And then Vivek invested in it.
Correct.
As part of his hedge fund.
Correct.
Okay. Okay, cool. This is it's very interesting, very helpful.
Yeah.
Changed the way I think a little bit, but should do more of these. Yeah.
Yeah.
Thanks, man.
Well, Sarepta is about to report earnings, so I'm going to go attend to that. Thank you, everyone, for listening. If you haven't heard, the FDA has hired a new curmudgeon that is going to stop all drug approvals and require p-values of 0.0001 with outcome studies for everything. Only overall survival will be accepted. So good luck, everybody. Have fun in the drug industry. See you later. All right. Why is HIMSS at 50? Problem 1. Why isn't Sarepta bounced back? Problem 2. I don't like losing money, but I don't mind future alpha either. So if you get a quick snapback on some of these, I wouldn't mind that.